Thank you all for listening in on this talk on the Biological Collections Ontology, or BCO. And I'd like to start by thanking my co-authors, John, Rob, and John, who made it possible to give this presentation remotely at the last minute. There's a lot of data available on biodiversity for biodiversity studies. We have great electronic resources like GBIF, EOL, NCBI, but it can be very challenging to integrate data from across these different resources, leading to what's often called a series of walled gardens. And the goal of the BCO is to reduce the height of these walls and make it easier to work across different data sources. The BCO, and here's just a link to our wiki page on Google Code, the BCO grew out of a series of workshops that took place about a year and a half ago, in which there was a realization that although existing resources like Darwin Core, ABCD, Mixis provide really wonderful vocabularies for describing biodiversity data, what they lack is the logical structure for reasoning over that data. And the BCO hopes to fill in that missing piece. So at those workshops, it was decided that the BCO would be developed as an ontology for biodiversity sciences within the OBO Foundry framework. So I'm just going to digress for a second to talk about what it means to be an OBO Foundry ontology. I think really the gist of belonging to the OBO Foundry is a shared commitment by a community really of ontology developers to create a suite of interoperable ontologies that span the biological and biomedical domains. The goal is to work for non-redundancy among these ontologies by reusing existing terms as much as possible to really have the most efficient development effort as possible, but also to make sure that terms can be used as widely as possible. When you have to coordinate with each other, it's much more likely that your terms are going to work for a larger group of people. Being in the OBO Foundry really just means an agreement to adhere to the OBO Foundry principles, and these include making your ontology available through open access, a willingness to collaborate with others so that you don't end up with redundancy, as I mentioned. There's some technical requirements like using a shared set of formats, relations, and naming conventions, and just some best practices like providing good documentation and a single person who agrees to answer emails so you don't end up with the situation where, oh, I thought she was answering it. No, I thought he was answering the emails. I think one of the biggest advantages to working within the OBO Foundry framework is access to the community resources. These include not only technical resources like tools for ontology releases, but expertise of many different ontology developers who are very generous and have been very willing to share their time to help create better ontologies. So although there's not a strict requirement within the OBO Foundry to use a particular upper-level ontology, many OBO Foundry ontologies do use the basic formal ontology, or BFO, as an upper ontology, and the BCO has decided to also use the BFO. And this graph just shows some of the upper-level classes of the BFO and some selected BCO terms. So as you can see, within the BFO, there are two primary branches: continuant, which includes things like materials entities, roles, qualities, functions, and occurrence, which for our purposes mostly includes processes. Within that framework, then, we have important classes in the BCO in orange include material sample and material target of observation, both of which are material entities, but which become samples or targets of observation through taking on different roles, such as the material sample role or the material target of observation role. Related to those entities are are processes, such as material sampling process. So for example, material sampling process has as its part selecting, physically extracting, and submitting some material entity, and it has as its output a material sample. As you can see, material sampling process is currently a subclass of the term planned process from the Ontology for Biomedical Investigations, or OBI, and we plan to have further integration with OBI going forward. For example, we will probably replace the term material sampling process with the OB term specimen collection, which really has the same, even though it has a different name, has a very similar meaning to what we intended. So although that slide focused on material samples, I want to just say that the scope of the BCO is slightly broader. It really encompasses three main types of biodiversity data collection. The first would be the collection of organisms and their parts, really what you think of as traditional museum specimens. That can include a whole organism. It can include a jar full of multiple organisms as a single specimen. It could include a herbarium sheet. The second class of data that we're really interested in is environmental or metagenomic samples. In fact, the linking of environmental samples to traditional museum collections was the original motivation for the BCO. In environmental sampling involves collection of some environmental material, water, soil, tissue of an animal, gut contents, etc., from which different environmental parameters are recorded, and then DNA is extracted to collect the metagenomic sequence. The third category of biodiversity data that's the focus of the BCO are surveys or inventories or other types of ecological observations. And the main thing that's different about that class of information is that often it includes data about the presence of some organism or taxon at a specific location, but does not include the collection of a material sample. What it might have in common with other types of sampling is this nested sampling. And nested sampling or tracking of data through different sampling chains has really been the focus of BCO development up to this point. So in order to describe, to sort of test the efficacy of the BCO at describing that nested or sampling chains, we created a small test case that's based on a real-world example. It's based on the Morea Biocode project, which was it had the goal of comprehensively sampling all plants, animals, and fungi on the Pacific island of Morea. We've just created our example to focus on insect collection. So you can imagine over the course of several years, various insect specimens are collected. Some of them are submitted to museums. Some insects are just photographed without the collection of a sample. And from those specimens, there may be a morphological identification based on a key. For other specimens, tissue samples are taken. Those would be sent out for sequencing, the sequence stored in GenBank, and then that sequence could be used as a barcode to identify the specimens as well. For other samples, for other insects, gut samples might be taken, and DNA extracted from those gut samples and a metagenomic sequence generated, which would be stored at yet another repository like the camera portal. So this figure just shows graphically how the BCO is used to model the kind of data that was shown in the previous slide. So in this case, the ovals represent classes within the BCO, and the rectangles represent instances or individuals. The color coding of the individuals corresponds to the color coding of the classes. So down the left side, the gray rectangles are all instances of the BCO material sampling process. And the way that we would use the ontology to track data through these various processes is, for example, we start with the original process, which would be a biocode sampling event. It has as its output an insect specimen. Then that insect specimen is used as the input to a tissue sampling process, and it has as its output a tissue sample. The tissue sample is in turn used as the input to a DNA extraction, which has as its output DNA molecules, or a DNA sample. Then that DNA sample can in turn be the input to a sequencing process, which has as its output the GenBank sequence. That GenBank sequence is used as the input to identification process, which has as its output the name of a taxon. So we created a sort of small, medium-sized, I guess I should say, data set of different processes, and here's a selected example of some of the selected processes. This is not all of them, with their identification numbers, the type of process, that is which class they're an instance of, their inputs and outputs, and some other metadata like the date. This is additional. We also created instances of a number of material entities and information artifacts. So for example, insect 1 is an instance of the type organism or virus or viroid. You see this column inferred type because the reasoner is able to infer that because insect 01 is the output of a material sampling process, it is in fact a material sample. So once we have all of the data into the ontology, we're able to query it to get out information that we would be interested in. For example, we can say, list all the processes that took place in 2010 as part of the Morea insect inventory. This would, of course, need to be done using Sparkle or something similar, but since I'm not a Sparkle expert, I just wrote it as a simple text using abbreviations for the ontology terms, not their actual URIs, which you would have to use. And so in this case, we get all these examples. These are all the different process. All of these process types are subclasses of process, so the reasoner knows to return them, and it knew that they fell out, that the date was 2010. You could do a slightly more complicated query, for example, list the output of every taxonomic identification process that has as its input insect 03, this organism. And in this case, we would get the first case would be fairly straightforward. You have a process by keying which has direct input insect 03 and directly outputs the name insect taxon 01. And the other result is slightly more complicated because the input insect 03 actually goes into this material sampling, tissue sampling process. But through a series of different sampling inputs and outputs, finally in this chain ends up with this output of an insect taxon name from this taxonomic identification using DNA barcoding. And you can see that these two different processes actually gave different outputs, and so that gives us a kind of a check on to know that something that we're having inconsistencies here. And you can imagine on larger data sets, this kind of reasoning could be used for quality control and checking inconsistencies in data. So just going forward, we hope to, in the technical end, we hope to set up a SPARQL endpoint soon with the example ontology, and so you can run queries of your own. It's not ready yet, but please check back on the BCO wiki page. When that's ready, we will put up an announcement and send that out on our mailing list. We also hope to implement some community curation tools like QuickTerm templates or BioPortal, but if you have requests right now, they can go to our issue tracker on our Google Code site. In terms of the ontology development, we hope to have some better integration with OBI and other ontologies, some more sophisticated treatment of naming, taxonomy, and identification. We want to do ontological modeling of surveys and inventories, like Rob is going to talk about in a second. And of course, we need to do mappings to Darwin Core, ABCD, Mixus, and others so that we can continue to test the BCO with some real-life data sets, some historical data. And with that, I'll just thank all of the people who've contributed to the BCO over the past year and a half, and the various funding efforts funders who have contributed to it. And if you have any questions, please put them on the BCO Discuss mailing list, the address for which is listed here. Thank you very much.